Good afternoon, everyone. Welcome. On behalf of the staff and affiliates of the Center for the Study of Adolescent Risk and Resilience, I welcome you to this talk by Danielle Dick. Dr. Dick is the Greg Brown Endowed Chair in Neuroscience, as you can see here. She's in the Department of Psychiatry at the Rutgers Robert Wood Johnson Medical Center, and she's the inaugural director of the Rutgers Addiction Research Center. Many of us know her for her work prior to moving to Rutgers early this year when she was at Virginia Commonwealth. She was the founding director of the Center for Research on Behavioral and Emotional Health and the founding director of Spit for Science. In fact, several of us in the room know her for that work, which ultimately recruited about 10,000 Virginia Commonwealth students into a longitudinal study in which they spat for science. They provided saliva that was then used to extract genetic data and to study essentially substance use and their emotional health during the college years and following. Her highly productive and influential research program focuses on genetic and environmental influences on behavior, specifically developmental patterns of substance use and emotional disorders, conduct problems, and how we can use information to inform prevention and intervention. Her 350 publications have been cited more than 30,000 times. She's been principal investigator on 15 NIH grants. This is something you might know about her. She has a 2021 book called The Child Code: Understanding Your Child's Unique Nature for Happier, More Effective Parenting, which is published by Penguin Random House. Just because it's fun to hear a couple of snippets from online reviews of this book by actual parents. A must read for any parent. It's a logical, science-based approach that makes sense. Another one said it's like a page turner because the writing is so accessible and engaging. For anyone who has ever felt attacked with parenting advice, judgment, you need to buy and read this book. High praise indeed for her work and evidence of her skill and ability at translating some serious science to actual applications. So please join me in welcoming Dr. Danielle Dick.
Well, thank you all so much for inviting me to come down and visit with you all. It is such fun to be back here after several years since my last visit. But some of you may know that one of my very first grants was a collaboration with Ken Dodge and other folks here when I was a first-year new faculty member at WashU St. Louis. So this has always been a place near and dear to my heart. So I want all of us to imagine for a moment that we all have our genomes sequenced. And you can know how at risk you are for hundreds of medical disorders, mental health outcomes, your disposition toward impulsivity, life satisfaction. Would you want to know? What would you do with that information? How might it shape your life choices? Would you want to know for your children? For some of you, this might feel very much like science fiction. But I want to take you back less than 30 years ago to an episode of the Today Show, where they were talking about this crazy new concept called the internet.
Back now at 56 past, I wasn't prepared to translate that as I was doing that little tease.
Oh, that's
that little
right.
mark with the A and then the ring around it.
At.
See, that's what I said.
Mm-hmm.
Katie said she thought it was about.
Yeah. Oh.
But I'd never heard it.
Around.
I'd never heard it said. I'd always seen the mark, but never heard it
Yeah.
said. And then it sounded stupid when I said it. Violence at NBC.
I heard be around
big
or
fight
about
up in the lunchroom the other
See,
week.
there it is. Violence at NBC. GE.com. I mean,
Well, Allison should know.
what
do you
is
say to that,
internet
Allison?
anyway?
Internet is that
Massive computer
right,
network.
the one that's becoming really big now.
What do you mean? That's big. How does one? What do you write to it like mail?
No, a lot of people use it and communicate. I guess they can communicate with NBC writers and producers. Allison, can you explain what internet is?
Think about that. Less than 30 years ago, what is the internet? Now we have an entire generation that's growing up without ever remembering a world where you didn't have instant access in your pocket to anything that you might want to know. It has changed the way that we vote. It has changed the way we book hotels and taxis. It has quite literally changed our lives in ways that were totally unpredictable 30 years ago. What's made that possible? What has contributed to these massive and unpredictable changes in society is what's been referred to as Moore's Law. It was named after Gordon Moore, who was one of the Intel co-founders, and he observed in the 60s that the amount of transistors they could put on a microchip, on essentially a square inch, was doubling every year. So it was leading to exponential growth. And that's what led to these really rapid changes that we have seen. Now, the white line up here is indicating Moore's growth. It looks linear because if you look at the y-axis, it's an exponential axis there. The white line is Moore's law. The green line is the pace at which genotyping technology is advancing. The field of genetics is the only field where the technology is advancing at a pace that is outpacing computer science. The basis of the precision medicine is largely rooted in genetics. So the idea that right now, when do you go to your doctor? When do most of us go to the doctor? When we're sick, right? Not, maybe we go for our annual preventative care, but the vast majority of doctor visits are when something is wrong. And then there's this process of trial and error where you're trying to figure out exactly what it is and then figure out what the best treatment is. And that is incredibly costly to the individual who's suffering, to their family, to our healthcare system. And the idea is that our genotypes contain a lot of information about what we are potentially at risk for, and we should be able to harness that to move medicine from a treatment-based model, you show up when something has gone wrong, to a model that is more predictive, personalized, and preventative. So we're essentially talking about how can we revolutionize the medical field. This was actually, so it was presented by President Obama in his 2015 State of the Union address. It was then signed into law with bipartisan support. When does that ever happen anymore? So it is billions of dollars that's going toward genetic research. A lot of it is going toward the collection of the All of Us cohort. Some of you might have heard of this. It's 1 million individuals being recruited at medical centers all across the United States with extensive phenotypic, biological, environmental information. Genetics is the second most funded area by NIH, more than $10 billion a year after clinical research. It's not just individuals who are in science and medicine who are interested in genetics, but in fact, there's also been an exponential uptake in direct-to-consumer genetic testing. So individuals that are going to private companies to get their genetic information. Not only that, but you can right now upload your raw genetic information, the output that comes from 23andMe, Ancestry.com, et cetera, upload it to free publicly available websites. ImputeMe happens to be one of them. This is the picture from the homepage. Interestingly, has a family on it there. And there is also, as you can see, been exponential increase in uptake of these of individuals who are directly accessing their polygenic risk scores. So on ImputeMe, you upload your data, and you can then select from more than 1,500 medical outcomes to request your genetic risk score. Half of the top 12 most requested genetic risk scores out of more than 1,500 conditions are for psychiatric and substance use outcomes. Alcohol use disorder, where a lot of my work is the third most requested medical outcome from ImputeMe. Now, if you're sitting there thinking, okay, well, I don't do medical research. I'm a social scientist. Genetics is coming to your area too. This is the future. It might not feel like it sitting right now, our brains have evolved to think linearly. And if you haven't seen it, I highly recommend talks that Peter Diamandis gives. He talks about the mystery and power of exponential growth. Our minds think linearly. We cannot comprehend changes that happen on an exponential growth curve. We couldn't comprehend the changes that 30 years ago, what's the internet, were coming. So this is the future. And I think the question is not, is genetics going to play a role, but really, what do we do about that? And so, to my mind, this really underscores the kind of research that many of us do, contributing to thinking about what are risk and protective factors, what are the trajectories by which risk and resilience unfold, become very important. And really what my work does is it attempts to integrate genetics into psychology and think about these big questions. And so my work really spans the translational spectrum. And what I'm going to be doing is talking a little bit about each of these areas today. So I lead big gene identification projects where we're trying to identify specific genes involved in substance use and related outcomes. And then I'm involved in a number of longitudinal studies where we're following kids across time and asking, what do kids who are carrying at-risk genetic dispositions look like as they're growing up? And importantly, what environments are either exacerbating or reducing risk? And then ultimately, what we want to do is use all of this information to develop more tailored and effective prevention, intervention, and treatment. A thread that runs through all of my work is also thinking about how we can train and diversify the next generation of researchers. Genetics has a real diversity problem, and it's something that a decade ago no one was talking about. Now people are very concerned about and thinking about from a health equity perspective. I'm not going to be talking about that today, but if there's time in the question period and you're interested, ask me about it. I'd love to. So I'm going to start really at the beginning, at efforts to identify genes involved in substance use and related behavioral phenotypes. As I mentioned, much of my area focuses on alcohol use outcomes. And of course, one of the things that we know if you're interested in substance use outcomes is that they are frequently comorbid. So alcohol with other drug problems, as well as antisocial behavior, conduct problems in kids, as well as other personality measures related to self-regulation and impulsivity. This is all seminal work that many of you in this room have contributed to. And what we also know is that there's not just phenotypic clustering, but we know from twin family studies, and this has been robustly demonstrated across lots of studies now, that part of what contributes to this comorbidity that we see, or this overlap, is that they are all influenced by a shared underlying genetic disposition. Often this is called the externalizing spectrum, and it's thought that this underlying shared disposition is something related to behavioral under control or disinhibition. You see different words being used. In the area of alcohol use outcomes, for example, and other drugs, we know that about 2/3 of the heritability of alcohol use outcomes is via this shared genetic disposition that also impacts all of these other phenotypes related to behavioral under control. That only a smaller part of the genetic disposition toward alcohol use outcomes is specific to alcohol. So one of the things that I've been trying to do with many others as part of this international consortium is to harness this information about what we know influences these different substance use and behavioral outcomes. So what we've learned about the ways that genes are influencing these outcomes to improve our ability to identify genes. And that led to launching the Externalizing Consortium. So it turns out that underlying latent factor is far more heritable. The heritability is about 80%. So when you move from the individual disorders themselves, which are heritable at about 50%, to that underlying shared genetic risk, it appears to be highly heritable. And so what we wanted to do was capitalize on that, to identify genes involved in this shared underlying liability to all of these behaviors and disorders, characterized by self-regulation or low self-regulation, and then identified genes that were also specific to the individual disorders or outcomes. We wanted to increase the accuracy of the genetic scores that we can create to index liability for individuals. We wanted to advance the underlying biology. I'm not going to talk much about that today, but when you find genes, you can also look at what are the gene circuits and networks that are represented, and then we pass those off to our animal model colleagues, and they work on how can we manipulate those and think about drug development, et cetera, and enhancing pharmaceutical treatments. But we also want to be able to characterize the pathways by which genetic risk unfolds across the life course. So the work that I'm going to be presenting in this first part was published in Nature Neuroscience actually last fall. This is the group of individuals that played a leadership role. And so if you're interested in any of the details, you can find more of the information here. Broadly, what we did is we essentially went out and looked for big GWAS, big genome-wide association studies that existed for phenotypes that were related to the externalizing spectrum. And so there were 7 that we identified. You can see them listed here. We were looking for samples where there were 50,000 or more individuals included, so powerful samples. And you can see we have ADHD, alcohol, cannabis outcomes, risky sexual outcomes, risk tolerance, smoking. And then what we did is we fit a structural model, but instead of now looking at phenotypic variance, we're looking at genetic variance across these outcomes, because what's going in is not an individual's phenotype, but individual's genotypes, the GWAS results for each of these outcomes. And we again identify a single underlying genetic factor, which maps onto the twin data. So there were 1.5 million individuals included in this data set. And what we then did was, rather than focusing on the individual outcomes for GWAS, for a gene identification study, like so much of the literature out there does, and partly that's a product of the way that our funding agencies work. And so there is a GWAS for alcohol outcomes, and then there's one for opioid use, and then there's one for cannabis, and one for ADHD. Well, we were moving essentially gene identification away from the individual disorders and to that underlying liability. So we're running a GWAS on that latent factor. And for those of you that do not eat, sleep, and breathe this world of genetics like I do, what we're essentially doing when we're running a GWAS is millions of regressions across the genome. So we have genetic markers, so SNPs, single nucleotide polymorphisms, places where people's genotypes differ, all the way across the genome. And we're essentially walking through the genome to ask, are individuals who are affected, are they more likely to have a particular version at that location? Or for a quantitative outcome like we're looking at, is it associated with higher scores on that outcome? We then do a massive multiple testing correction, as you might imagine, since this is an atheoretical agnostic test. And this produces what we call a Manhattan plot. The idea is that you want it to look like Manhattan. You want it to look like there's lots of skyscrapers and peaks there, because what every single dot on there represents is one of those millions of markers that we tested. And essentially, the line here is the line of significance after taking into a multiple testing correction. So everything that is up here, the little dots that are up there, are all significant at less than 10 to the -8. And what you can see is that we identified 579 conditionally independent loci. So 579 different locations across the genome that were contributing to this underlying latent factor representing behavioral disinhibition. Compare that for a second to the biggest GWAS that were out at the time for alcohol use disorder. They identified 29 loci. Or cannabis use disorder, 2. Opioid use disorder, 1. And I think this really powerfully speaks to how when we move gene identification away from individual disorders, and we use what we have all learned about clustering of behavioral risk, and instead are thinking about how genes are influencing some of these underlying factors, It's far more powerful. So what we can then do is take these results, and for every single one of these loci across the genome, for every individual in our data set, we can sum up how many of those risk variants they carry, weighted by the effect size associated with each one. And that creates, for every individual, a polygenic risk score, sometimes called a PRS, or increasingly people are moving away from this term to PGS, just for a polygenic score. Because sometimes for some of the outcomes we're talking about, you might not, if you're thinking about personality, I don't know, are you at risk for extroversion? So it's all the same method. You are essentially summing up to create a polygenic score for each individual, indicating their liability on that outcome. So we then went into two independent samples. The COGA sample, this is a longitudinal study of families that have been enriched for alcohol use disorder. Someone in the family was recruited through a treatment center, and then we essentially tried to fill in the families and go down a generation. So we were looking at, we had data from over 12,000 individuals from more than 2,000 families. And we also looked at the ad health data, so nationally representative sample from the US. And what we found is that when we calculated the PRS in those independent samples, and we created an externalizing factor score with the same variables that went into our original gene identification, or similar ones that were found in those data sets, it accounted for about 10% of the variance in both data sets in externalizing outcomes. And this is now getting to the kind of variance that we see associated with many of the social, environmental major factors that we're interested in the social sciences. But not only that, it was not just associated with the externalizing outcomes that went into the analysis. It was also associated with a host of other behavioral, socioeconomic, and other kinds of important life outcomes. And so what I'm going to show you is these are data both from COGA and Ad Health. And to orient you to these graphs that you'll see, we have essentially broken up genetic risk. They're, of course, continuous. The polygenic risk scores are continuous. They're shaped like a bell curve. They're distributed just as we might expect in the population. We broke them into quintiles. And so at the far left, you have individuals at the lowest level of genetic risk. All the way to the far right here, we have individuals at the highest levels of genetic risk. And what we mapped is the percentage of the sample that, for example, on the far left, you can see that that top line is for moderate AUD. And so individuals at the lowest levels of genetic risk have about a 30% lifetime risk of developing AUD. Individuals at the top have like a 50% lifetime risk of developing AUD. Ever use of opioids, individuals at the lowest levels of genetic risk have about a 15% chance. Individuals at the highest levels of genetic risk have closer to a 30% chance, so doubling of risk. Not only that, they don't just predict substance use outcomes, they predict things like ever been arrested, conduct problems in kids. And I should mention for a moment here now that all of the individuals who went into the discovery analysis and who were used in these independent replication cohorts were individuals of European ancestry. So we don't have any of the complications that you would expect in the United States with respect to racial ethnic discrimination that exists in our criminal justice system, et cetera, taken into account in some of these lines when looking at things like arrests. It also predicts things like college completion. So individuals who have the lowest levels of genetic risk, so essentially good self-regulation, you can see nearly 50% of them completing college. Individuals at the highest levels of genetic risk, lowest levels of behavioral control, only about 20% of those individuals complete college. Being fired from work. So you can see a whole host of really important life outcomes that are related to one's genetic liability. We then took these scores into electronic health records using the Vanderbilt Biobank. So we had more than 1,500 biomedical outcomes. And though you can't see all the details here, now what all these dots represent is instead of in a GWAS, we are essentially have one phenotype externalizing, and we're scanning markers across the genome. Now what we have is one genetic risk score, and we are scanning for those individuals, what is that genetic risk score correlated with across all of these biomedical outcomes in their health records. And what you can see is that polygenic risk scores related to behavioral under control. So again, everything above this line, all these dots are significant. It's associated with substance use outcomes and psychiatric outcomes and things like suicide attempts, as we might expect, but it's also associated with worse health in nearly every bodily system. And it's most strongly associated with things like heart disease, HIV infection, type 2 diabetes, obesity, cirrhosis of the liver, lung cancer. All of these are medical outcomes that are mediated by behaviors related to self-regulation. Drinking too much, smoking too much, risky sex, eating too much. So a genetic risk associated with decreased behavioral control is actually associated with a whole host of important life outcomes that go far beyond substance use and the psychiatric outcomes that we started with. Now, maybe at this point you're thinking, okay, but substance use outcomes, for example, or psychiatric outcomes, we know are associated with socioeconomic status, and then that's going to be associated with college completion and all of these other kind of things. So maybe there's just tons of confounding access to the healthcare system. That's why you're seeing everything being correlated. So we then looked within siblings, and we did it in 3 independent samples: siblings in the UK Biobank, siblings in COGA, and siblings in AdHealth. And we asked, okay, now within a sibling pair, so you're holding all those family things constant, and we're asking, is the sibling who has a higher genetic risk score, is that still associated with these outcomes? The things where there was the greatest attenuation, as you would expect, are for things like cognitive ability and the socioeconomic outcomes. But for substance use, psychiatric outcomes, all those other health outcomes, the results are attenuated just a tiny bit. They are still strong and significant within families. So we don't think that what we're picking up is purely confounding or other sorts of population stratification kinds of issues. So what I think these results tell us is that when we move away from gene identification and thinking on a very disorder by disorder, and we think about what do we know about the underlying risk, it can really advance our gene identification efforts. This is now one of the most predictive polygenic scores to date. And of course, gene identification is just the first step. Then the really interesting things, the questions that we can ask, now become possible. And I'm going to move to talking about some of those next. And so we know that there's many ways that genetic risk might influence an outcome. So maybe there are direct genetic effects, meaning that these are genes that are influencing the outcome directly. So for example, for alcohol use outcomes, we know that there are some genes that influence the way the body physiologically breaks down ethanol that can contribute to risk. I would think of that as a kind of direct genetic effect. It's influencing a biological process which impacts the outcome. We also know that the environment can change the association between genetic risk and the likelihood an individual will have an outcome. So for substance use outcomes, if in fact, for environmental, religious, societal, et cetera reasons, you never use alcohol, it doesn't matter what your polygenic risk score is, you're not going to develop an alcohol use disorder. The environment has changed that out, the likelihood of an outcome based on genotype. But we also know that our genes influence our selection into environments and the way that people respond to us, and that those intermediary environmental pieces can also contribute to the outcome. And so one of the things that we've been working on, and these are analyses that we did again in the COGA sample, in a younger subset of that sample, 1,200 adolescents, and I'm going to be presenting data following them at 2 time points in adolescence, at 13 and at 15. And we were interested in this interplay, in this case, between polygenic risk scores, associated with externalizing outcomes, and parental monitoring and peer substance use. And so this is the model that we fit. And so we're allowing for direct effects of the child's PRS, the genetic liability on externalizing behaviors. It's essentially a factor score of the things that went into the original analysis, conduct problems, substance use. And then we're also testing whether at age 13, already above and beyond the association with externalizing behavior, there's any association between genotype and the child's relationship with parents and their relationship with peers, and then how those things play out across time. And so, first of all, to orient you to the blue arrows highlighted here, as expected, we do find direct effects of the child's genotype. There's an association with externalizing behaviors at 13, and then that influences subsequent externalizing behaviors at 15. But we also see, in addition to the correlation that exists between externalizing behavior and parenting, we see additional effects of the child's genotype on their relationship with their parents. In particular, kids who are carrying more genetic risk, so these are the more impulsive kids, less regulation, have reduced parental monitoring. I have one of these kids. They wear you down, right? And we're already seeing that at age 13. Then, decreased parental monitoring subsequently is associated with selection into riskier peers, which then is influencing their externalizing. We also see that child's genotype, above and beyond its association with externalizing, is impacting selection into riskier peer groups. And then those riskier peer groups are impacting future externalizing behavior, again, above and beyond the child's own externalizing behavior. So when we put all this together, we find evidence both for direct genetic effects, but also for these indirect pathways via environmental risk factors. So we're finding that kids' genotype is also influencing relationship with their parents and selection into riskier peers. So we see these essentially cumulative patterns of risk that have been documented in other kinds of longitudinal studies, but we can show that it is associated with the child's genotype. So there's evidence of direct genetic effects, but the piece that many geneticists forget when we find these associations between genes and an outcome is that some of that association is a function of environments that the genotype is influencing, which is impacting the likelihood of that outcome. And then finally, what are we going to do with all of this information? Ultimately, what we want to do is to use it to inform prevention and intervention. And this brings us to, I think, the question that we have to start by asking, which is, if we're talking about using polygenic risk scores, do people even understand what polygenic risk scores are? This is the output from Impute Me, that publicly available website where I said you could upload your raw genetic information and it's going to give you information about your risk. Now, I work in this field and I think that this is incredibly complicated and hard to understand. So one of my graduate students, Morgan Driver, who's doing a joint Ph.D. in genetics and master's in genetic counseling, we started by doing a review about what actually do we know about genetic feedback for psychiatric and substance use outcomes. Turns out there have been a number of studies which have looked at, are people interested in getting this information? And then we also did studies in our college students. Consistently, 80% of people say, yes, I want my genetic risk score for psychiatric and substance use outcomes. But concerningly, across all these studies, we always find about a quarter to a third of individuals misunderstand the role of genetics in these complex outcomes, meaning they think of genetics as determinative. They think it's single genes, or they think if you're at genetic risk, the environment, you're destined to develop this problem, that the environment doesn't play a role. And not only that, there's also some suggestion that receiving genetic information that indicates an increased risk of this particular challenge negatively impacts affect, lowers an individual's confidence that they can do something about the behavior, and reduces self-agency. So in other words, it's the exact opposite of what we are hoping to achieve by integrating genetic information. We want it to be empowering for individuals to be able to use this information to make the best choices for themselves. And there's evidence that, in fact, the exact opposite could happen. So, work that Morgan did as part of her dissertation, we did a randomized clinical trial to evaluate the impact of educational information, which we built with data visualization specialists. They were out of the Broad Institute at Harvard. And it explained polygenic risk scores, essentially. All of these concepts of it's lots of genes, here's what it means, the environment still plays a role, here's what you can do about it. And then we essentially assigned people either to get that information or to just get information about alcohol use, understanding alcohol use. And then we looked at whether, in fact, it did have an effect on people's understanding. And so the good news is that we found that this a very brief online educational program, it does increase understanding of polygenic risk scores compared to other conditions, and that participants indicated that when they got increased levels of genetic risk, we randomly assigned people to get different levels of hypothetical genetic risk. Imagine this is your genetic risk score. That they reported intentions to reduce the drinking when they received elevated genetic risk. We did not find the opposite, which is the question everybody asks, meaning what happens if you get a low genetic risk score for substance use? Will young people be like, woohoo, now I can drink as much as I want? We found no evidence of that people would report increases in their substance use. But it turns out we don't even have to wait until we have found all of the genes and people have genetic risk scores. We know already that for these complex outcomes, both genes and the environment are important in an individual's outcome. But very frequently, when we think about prevention and intervention, we're focused on the environmental side for very good reason. Obviously, that's a critical piece that we want to address. These are challenging problems, and we're essentially leaving the other half on the table. We're not fully making use of all of the information that we have about what contributes to risk. And so we can incorporate this information about what we know are these intermediary phenotypes that represent early behavioral indices of genetic risk and incorporate those into prevention and intervention as well. And so I'm going to end by talking a little bit about an effort that we led in our Spit for Science project, which Rick mentioned earlier. This was a university-wide research project focused on substance use and mental health in college students. We enrolled 5 cohorts, so we have DNA on more than 12,000 individuals. We got about 70% of all incoming freshmen in the years that we enrolled. And part of the idea was to use the findings to feed back to develop prevention and intervention for the university community. We know that college students engage in high rates of risky substance use. We also know that they are highly unmotivated to participate in anything that even sounds like substance use prevention programming. Universities have all kinds of creative ways of getting around this. At VCU, some years we wouldn't allow them to register for classes until they had finished their mandatory alcohol training. Many universities do that. One year when it wasn't mandatory, we found that only 40 out of 4,000 incoming freshmen even clicked on the link for the information about substance use. So what we've been thinking about is how do we essentially find creative solutions to this problem? Or what the mom in me calls chocolate-covered broccoli. In other words, how do you get something that's good for someone to them in a way that they actually will enjoy and want to be part of? And so this led to the development of what we call the personalized feedback program. Turns out college students don't want to have anything to do with substance use prevention programming, but what they do love to do is fill out quizzes about themselves. So you see this on social media, what superhero are you, what dog do you look like, etc. So we were like, let's capitalize on this. So we called it Me Time, learn about yourself to get the most out of your college experience. These photos throughout are the team that has led these projects. They filled out about 20 questions about themselves. And of course, what these questions were tapping into all of the things that we know are related to substance use outcomes. So questions regarding risk-taking, regarding impulsivity, regarding anxiety sensitivity, extroversion, which in college students we know is also associated with riskier substance use. They filled it out, they get a little bit of psychoeducation about how our genes and our environments come together to impact our outcomes. They could then create their own avatars. We essentially worked closely with, we did a lot of focus groups with students. VCU is a very diverse undergraduate population. We made sure that we had all kinds of skin tones, hairstyles, et cetera, recommended. And so they get their profiles both in a kind of quantitative way across these 4 dimensions that we included, which were essentially sensation seeking, extroversion, impulsivity, and anxiety sensitivity or neuroticism. And so they both kind of got a binary assignment. We worked with our clinical service providers on using language that we felt was acceptable to students. And then what we essentially are doing is integrating the literature on genetic and personal risk factors with CBT and harm reduction principles. So for each of those outcomes, we would walk through the good, the not so good, and level up. We would connect them to personalized resources associated with their particular risk profile. So we run a small RCT. We find very high satisfaction. So 85 to 90% of students report their personality profiles look accurate, they liked it, they had fun, etc. They also report intentions to use more resources after finishing the program, and that they intend to use fewer substances. And when we then followed them up 30 days and 3 months later, this was all happening during COVID. So as we know, this was a really interesting natural experiment to show how the college environment is a very risk-enhancing environment. Rates of substance use were much lower than when they are on campus, when they were home. So essentially, what we were also looking at is not just reductions in risky users, but we're trying to keep individuals keeping low-risk drinking patterns. And what we found is preliminary evidence that the PFP, when compared to the sort of standard brief motivational intervention, which provides feedback on alcohol use patterns and harm reduction strategies, and a control group, and even a combined group where they got the PFP and the brief motivational interview alcohol-focused program, that the PFP was the one that was associated with reduced substance use 30 days and 3 months later compared to the other ones. So I think some indication, even more so than the combined program, which I think gets at, for all the parents out there, we know you can't talk to your kids too much or else they shut out and they shut off and they tune out all of it. I think there might be some element of that going on here where it's kind of the just right dose. And of course, what we're trying to do with all of this is take the child who we can see impulsivity show up at a very young age. It's the child that's dangling from the tops of trees and giving their parent a heart attack and ending up in the ER a lot. And we know that that child is at greater risk of, fast forward a decade, becoming this person. But not necessarily. There's no reason you can't channel that disposition into this or into this. And for full disclosure, this little risk taker is my little risk taker who comes by it very honestly. My father, his father, my brother, all fighter pilots, an alternate way to channel a risk-taking disposition. So when we put all this together, I think genetics is poised to shape the future in ways that are likely to be far-reaching and that we can't even imagine right now, due to the exponential growth that is happening in this field. And so I started by showing you a clip from the mid-90s of the Today Show, where they were talking about this crazy concept called the internet. I want to take you back to the mid-90s to one of my favorite movies, Gattaca, came out in 1997, a science fiction movie.
10 fingers, 10 toes, that's all that used to matter. Not now. Now, only seconds old, the exact time and cause of my death was already known.
Neurological condition, 60% probability. Manic depression, 42% probability. Attention deficit disorder, 89% probability. Heart disorder, 99% probability. Early fatal potential.
Okay, so they clearly got some things wrong. You can't know exactly when you're going to die at the moment of birth. But think about the other parts. Manic depression, 20% probability. ADHD, 80% probability. Think about where I landed with our latest gene identification efforts. With this genotype, 50% probability of AUD, or 30% probability of childhood conduct problems. That was science fiction 30 years ago, less. Today, it's a reality. It's not widespread in our society in the same way that the internet wasn't 30 years ago either. But I do think it's coming. It's coming in ways that we can't even comprehend right now. And I think this makes it incredibly critical that we need to enhance understanding among the public and as researchers to be able to map those pathways by which genetic risk unfolds, how it interacts with the environment, and to enhance the public's understanding of these complex gene environment pathways. This requires us to step out of our comfort zones. Rick mentioned the book that I wrote. I call it my pandemic project. And really, it was born in my head years prior because when I had my son, I found myself raising the challenging child that I study. And I found the research to be so incredibly helpful. But for the first time, I was looking around at all these parenting blogs and magazines and books that were out there, and I was struck by how so many of the messages for parents didn't match what I knew about the research. Mainly, they're not at all talking, they're talking all about the environmental kinds of things parents can do, really important, good knowledge, but nobody is mentioning the genetics. And this can lead to, I think, inadvertently, a lot of pressure on parents, a lot of guilt if, gosh forbid, your child isn't behaving in the perfect way or they're struggling. What am I doing wrong? And really, that's what led me to write the Child Code, with the idea that parents need this information. They need it now, but they're really going to need it if we're thinking ahead to the years to come. Writing a trait book, I will tell you, I went from being a professor where I kind of know how this gig works to knowing nothing about how that world works. And if anyone's interested, I'm happy to talk about it. But it does require us to step outside of academia. We talk a lot about impact, but think about impact as more than just the journals we're publishing in. If we don't get out there and take our science to the public, someone, likely someone far less knowledgeable, I mean, who wouldn't want to take genetics advice from the rock, is likely to fill that space. So if not you, then who? I want to end by acknowledging the many amazing members of my team who have contributed to all of this work that I've presented today, our funding largely from NIAAA and from NIDA. I thank you for your attention. And if you're at all interested, I have become increasingly passionate about how we need to do more to get science out to the public. It requires us to write in different ways, to produce blogs and videos and social media and other kinds of different things. If you're interested, here's some of the places that you can find me as I attempt to voyage into this brave new world. I thank you for your time, and I'm happy to take any questions. Yes.
Thank you so much for your talk. It's extremely compelling. I wanted to ask about like the exclusivity of like the externalizing factor in terms of psychology, like you mentioned anxiety, depression. So do those look different for internalizing factors?
Yeah, it's a great question. So most of my work has been in externalizing, and we know that all of these things load closely together. So I also mentioned that so we can parse out what is shared across all of these disorders, which have elements of behavioral under control here. And then what we also get are residual genetic effects on the individual disorders themselves. What we find is that the GWAS results from that latent externalizing factor don't correlate strongly with depression, but the residual genetic effects on alcohol use outcome do. And I think that reminds us that anything that's not included in that model is not being taken into account, of course. So we probably don't think of genes involved in depression as genes that are specifically influencing alcohol use, which is how I conceptualized it. But what's ending up down in that residual are genes that are influencing alcohol use through depression, as well as through other kinds of psychosis, like schizophrenia is correlated more strongly in there. Separately, there are big gene identification efforts focused on depression and anxiety, so on internalizing outcomes. And then what we can look at is the genetic correlations between the genes we're finding for depression, internalizing outcomes, and genes for externalizing outcomes. They do show some correlation, as you would expect, but it's not super high. And we, in fact, put them into our models so that for any individual, we can look at both of those and we find independent predictive power. But the depression PRS, in our analyses at least, are not significantly associated with alcohol use outcomes in the way that the externalizing are, and especially not after we have externalizing in the model. Yeah.
Thinking of the impute need concept. So you're giving, these are unconditional priorities.
Yes.
Or, excuse me, risk factors or probabilities. Is there any advantage to giving conditional risks? Do you input conditional factors that we know moderate?
Absolutely. And so I'm so glad you raised this. In the world of genetics, we talk about relative risk versus absolute risk. And so what this is presenting is relative risk, right? So when we show a whole distribution of PRS, and then we're saying, okay, and you're up in the 90th percentile, that's your risk relative to the population. What we found is even with our educational materials and that RCT, people still missed that question, right? It's very confusing because they would think, oh my gosh, I have a 90% chance of developing alcohol use disorder. And so, there's the whole genetic counseling literature, because I collaborate closely with some colleagues in that field. There's really robust evidence that you shouldn't give people a relative risk, you should give absolute risk. And so that is where, and if you look at like what 23andMe does, it's not what impute me does. It essentially converts it to little diagrams with, 100 little figures, and they shade in, 10 of the figures, this is how many people with your genotype are affected as compared to 2 of the shaded figures in the population. So you're right, we absolutely need to do that. Yes.
Yeah, thank you, Danielle. This is terrific. I had 2 questions. Now I have 3 following up on Greg's. The kind of interaction effect that I mentioned, Greg's question was, so let's just imagine you've got a 9th grader who's causing all kinds of conduct problems, and the parents get the polygenic risk score, and it shows that the kid has a high % probability of being in jail. But if you could also say, but if you can get him to graduate from high school, his risk will go down to a lower %. It gives them either a goal or an intervention or something, or if you enroll them in this program, or if you, whatever, you can lower it to give them an idea of the power of the environment within this context of a fixed genetic risk.
Yes, and I'm sorry I misunderstood your question. I 100% agree. That's exactly what we need to do. And I do not think, there were questions earlier about stigma and what happens when you give people this information. I do not think you should give people this information and be like, you're at high genetic risk, good luck to you, because both we know that they will misunderstand it. But I think we also need to give the information, and I love how you presented it, to be able to quantify, okay, but now here are the other things you can do, and here is what that changes the risk to. I think of it as a two-part process, and this is, where I think companies like 23andMe fall short, which is that you have to, if you're going to give somebody a risk profile, you also need to connect that to research, to what they can do about it to reduce the risk. And so I think that those two pieces have to go hand in hand and are critical.
One more question. You very nicely talked about your family, your child, and all. In your empirical studies, are there any positive outcomes associated with the high externalizing apologetic risk?
So this is really interesting because it gets at, what is externalizing, what is underlying risk. And, many times we'll be talking about impulsivity and we're talking about different things. And so I know I'm preaching to the choir with this group who is so into deep phenotyping. And so for things, for example, like sensation seeking or risk taking, we actually know that CEOs and entrepreneurs tend to be higher on risk taking, fighter pilots, risk taking, sensation seeking. So one of the things that we talked about in that prevention program, and that I think is key, is that these are not bad things in and of themselves, right? There's upsides and downsides to everything. Even if we think about anxiety, there are some upsides to having a little bit of anxiety. It's what makes us prep for a talk or study for that test, or now too much can lead to challenges. And there's things, of course, that you can do to then reduce the likelihood that that's actually going to lead to clinical challenges. And so I do think that for many traits of interest, there's, thinking about it in terms of what's the good and what's the not so good associated with this. If you're presenting something like a PRS associated with ADHD, okay, then, even ADHD, I think you can talk about, and there's lots of literature out there about like, hey, here's some of the, great things about having some of these traits. But let's pick some medical condition that we might imagine there only being a downside to. I think that that's where presenting that information, but in the context of this is not determinative, and here's the things you can do to present to reduce risk. Even if there's not an upside of having that genotype, to know that it is not destiny, and here's what you can do is critical.
All that makes logical sense, and I'm just curious about whether within your empirical studies you measured any positive outcomes that might be associated with it would give empirical support to the description.
Yeah, so I know if I could say like, oh, you've got a 50% chance of being a CEO, and the funny thing is, we don't usually measure that stuff. Maybe you all do a better job, but a lot of the longitudinal studies that I work on, they're funded by the National Institutes of Health, and we tend to, I came out of a clinical program. We measure a lot of psychopathology, and we have historically, now there's of course a lot more focus on well-being, but a lot of the things that we're interested in are certainly not well measured in many of the data sets I work on, which doesn't allow us to, I think, we're so focused on what are the risk factors or what kind of things promote resilience for these adverse outcomes, that I just don't think we do as good of a job. I think we should, and if you have some of those data, let's do that.
Yeah, it'd be good to accumulate.
It would.
As a concrete example, my son has learning disabilities and he went to a special school for learning disabilities. The headmaster gave an introductory address that he said was based in his experience, but it's probably not empirical or systematic. But he said to the kids, you have a learning disability, and unfortunately that means that you have a higher than average risk you're going to end up in prison. He said, but my experience also is that you have a higher than average likelihood that you'll become a millionaire. I don't think he had any data to support that. But the concept was compelling.
Yes.
To have these kids characterize themselves as neither good nor bad, but it's kind of to be determined which way they will go. And that's what you were implying. Your son could either go to conduct disorder or become a fighter pilot.
I'm pleased to report that at 15 and a half, he actually is a straight A student and doing quite well. So the child who, in second grade said to me, Mom, school's just not really my thing. And I said, well, presumably you don't want to live with me for forever, which means that you're going to want to either get a job or go off to college, which means you need to learn these skills. And he said, I'm in second grade. It's not going to have any impact on where I go to college. I'll worry about it when I get to high school. And darn if the child who didn't barely get through 8th grade, suddenly was making straight A's in 9th grade. And I went, okay. But it does speak to that. For me, who was the straight A overachieving student, why would you not apply yourself? It was actually, if you want to think about it, a pretty adaptive way of being I'll put in what I need to, and then when it matters, okay, now I'll spend some additional time on it. So differences and outcomes.
Given this potential interpretation, you can see why calling them polygenic scores.
Exactly.
Is wise.
And honestly, I just haven't changed all of my slides yet, but we certainly are moving toward polygenic scores as opposed to polygenic risk scores for exactly that reason. They came out of originally medical terminology, and the first UOS were all thinking about it in terms of schizophrenia, bipolar disorder, alcohol use disorder, so it was risk for an outcome. And of course, now what we all know is that it's far more complex. Yes, Sarah.
I have a question. This is related to your intervention work and thinking about how you communicate with kids on a level that is a little bit more understandable. And I think that the feedback program is really an exciting way to do that. But I'm curious about, this might be a long-term question, messages that you might give in terms of drinking and the environments that college students might be exposed to when they're a freshman versus when they're a senior. More specifically when they're under drinking age versus at drinking age, the differences in how they're getting access to those things and even just their attitudes toward it, how it might change. So I'm curious if that's something you've thought about in terms of if you followed up with students after freshman year, sending messaging. Yeah, teasing apart those differences.
So it's a really interesting question. And the program that we developed was with the idea of incoming students. We really don't address this issue of it's illegal versus, now you're of drinking age. And I also want to be clear, I actually am, other than when I'm giving talks or interacting with students, I'm not doing any of the clinical work with students, but I worked very closely with the director of who had run our substance use programming at the university for decades. She retired while I was there. And she was amazing. She was that person that the students, she had just the right degree of authority, so they listened to her, but she got them and they got her, and it was just, she was amazing. And when she would talk to the students, she would essentially say, if I were queen, I would do away with the idea of a legal drinking age, because we know that that's influenced by politics and all kinds of other things. But instead, it would be like driving, where you get a permit, right? So all of you all have showed up. You are here now foraging as adults, and so you got your permit. And when I walk into a party, or the party patrol, the police did that, go into a party, if you're real drunk or you're doing things that are ridiculous, you've lost your permit now. And now you're going to probably have to do some of that substance use programming and talk to someone and all those kind of things. But if you are enjoying a beverage with your friends, great, everybody go about your merry way. And so she said, so I talk about the idea that we are all every day making decisions based on, rewards and consequences. If you are under 21, you know that is a possible consequence. How adverse or severe those consequences are even varies by university. It varies by all kinds of things. So you're making those decisions and you're weighing that. And so that's one of the things you're going to have to weigh if you are under 21. But her emphasis was, I would think about it much more in terms of how can you develop strategies that are going to allow you to have the relationship you want with substances. And so hearing her talk to students about it that way has influenced the way that I think about it as well. And I'm blanking on her name. She has a podcast called Ask Lisa. And she wrote, she's written several books that have done really well. She's a clinical psychologist. Anyhow, she has a great podcast, and she talks a lot about talking with kids, and it's, for parents, how to talk to kids about things. And she has an episode where she talks about talking to parents about how to talk about alcohol use with kids. And it's very much about having a dialogue and helping them think through all of these kind of pieces. And then what are the rules in your household and why? And so I actually think that the question of the legal drinking age is maybe less important than some of these other questions about how you are weighing risk and consequences and those sorts of things.
We have passed the hour. I saw that Megan just chatted the authors.
Yes, Lisa D'Amore. Thank you. I tell you, this is what I love about instant access to technology.
Yeah, thank you so much, Danielle.
Okay, Jenny, do I need to do anything for your computer over here?